My name is Anna Ridderstad Wahlberg, and I work at SciLifeLab at the Drug Discovery and Development Platform, or short DDD. Today, I will tell you a little bit about who we are and what we do. SciLifeLab is a national research infrastructure with a focus on life science, and we are organized into different platforms with different expertise and technologies. One of those platforms is the DDD platform. The aim for us is to support academic research to take the first step in drug discovery. DDD has a mandate from the Swedish government. It has been around since 2014, and it is fully embedded into the SciLifeLab infrastructure. I will start to talk a bit about the drug discovery process, and this is the roadmap for drug discovery in a very high level, which you often see when you have it often looks like this in the big pharma context. But when you're an academic researcher or an early startup, you don't have a road which is paved in the same way. And you have to learn, what do I need to do, what is the most important things in the early stage, and how to get started. And this is where the DDD platform comes in. And our focus is really in this early phase. And we can support the researchers with setting up the assays, setting up methods and screening for identifying hit, going further to lead generation, and also to generate a prototype drug, which is one of the end results that you can have in the drug discovery and development platform. But going into a little bit more detail, some of the early questions you have to look at to be sure this is a good project idea is the target valid? Does it have a connection to the disease you're intending to treat? Are there any safety issues? And some other really important questions in the beginning. And another important question is, can we actually generate starting molecules? Is it feasible for lead generation? And this is both on setting up the right assays, but also on the intended mode of action. And can we actually set up to transfer the research assays to the more industry way of working and the way towards developing a drug molecule which can go further into the clinic. And those kind of questions we do discuss with the researcher. The end result from working with us can be a prototype drug. But it can also be a very good assay that you can develop further at another stage. As I said before, the SciLifeLab DDD platform has been around for about 10 years, and we have got very many project proposals, almost 500. And of those, we have selected around 80 projects that have gone into our platform. And the thing is that the project that start working with us, they can be in many different stages. Perhaps they have a new idea, perhaps they have come a bit, and they continue with us in the platform. And so the road for each project can be very different. The mean time a project spends with us is around 4 years, but it can be shorter and it can also be longer. And what you can see on this picture is how we are divided into different units. And each unit has its own expertise, instrumentation, and competence. And we are working with project ideas in several different modalities. And modalities, that means types of drugs. Long time ago, most drugs that came on the market were small molecule chemical drugs. Nowadays, around half of the drugs that are entering the market are antibody or protein type of drugs. But also other new modalities are coming up. It can be oligonucleotide type of drugs, and those are based on RNA or DNA molecules. And there can be other new modalities that are conjugation of different components forming the final drug. And for being able to support the researchers to take their journey, we need to have the different competences that can support the different types of drugs. And we are around 50 people divided into 10 different units. And each unit has all the relevant competence for what they work with. And we are also divided into 5 or 6 different host universities, as you can see on the slide here. And today we will talk about a special unit which is called Biophysical Screening and Characterization. And you will hear a little bit more about that by Annette, who is the head of this unit. And she will today talk about the role of biophysical drug studies in drug discovery and development. And one of the major questions here is, how do we really know that a drug binds its target?
Biophysics. What is this? In Cambridge Dictionary, it says the science that uses laws and methods of physics to explain biology. This is quite a broad concept, but it basically means we use physical methods to understand biological concepts, often in a test tube setting. Biophysical methods for drug discovery. Here we mean a special set of methods. By the way, my name is Annette Roos. I am a crystallographer and structural biologist by training, and I work for SciLifeLab Drug Discovery and Development Platform. Now, biophysical methods for drug discovery. We want to look at binding, especially. How do the drug molecules bind the target protein? Also, biophysical methods can be used for sample integrity. In binding, so how well do the molecules bind the target, and how do the molecules bind to the target? So how well is a measure of affinity, and how is actually, in atomic view, how does the molecule bind to the target? Sample integrity. Here, this is important because the molecules we work with have to be pure and stable, and they have to have the right form for binding, and this we can measure with biophysical methods. We are nearly always working with a protein target. Doesn't have to be a protein target, but quite often. And the protein has to be pure and homogeneous. Instruments for measuring binding. Here are the instruments that we have at SciLifeLab in my unit. So we have surface plasmon resonance biosensor technology, SPR for short. And this is what I will mainly be talking about in this film. Then we have something called switch sense technology. It's another pretty white box. And then we have something called microscale thermophoresis. And these instruments can all be used to measure how well does a compound bind to its target protein, or how well does an antibody bind to its target antigen. In a drug discovery program, you tend to set up a screening cascade for how the molecules are to be developed and how they're to be tested. This is a typical screening cascade that we might have. We do a first screening of a library of compounds, often, not always, but in an enzymatic assay. Then we want to follow up the hits that we generate in this first screening situation. These are followed up in the enzymatic assay, again, and in a counter assay to see there are no false positives. But also we want to have an orthogonal binding assay, and this is where SPR comes in. Further, to reiterate the chemistry, get better compounds, we can use crystallization of the enzyme together with these compounds, then actually see how they bind to the target. In this part, also, it's extremely important that the protein quality is good, and it has to be tested. And that's where these biophysical methods that can measure sample integrity come into place. This is an instrument we have where you can measure something called nano-DSF, which is differential scanning fluorimetry, also known as thermal shift assay. Here you have your sample, first at lowish temperature, 15 to 20 degrees, and then you heat it up slowly, up to 96 degrees, and you measure what happens to the sample while it's heated up. You can imagine that a protein will start unfolding and aggregating as it gets hotter and hotter. And that's what you see here, and you measure some kind of light. In this instrument, you measure fluorescence, the intrinsic fluorescence of the protein. And here you see something happens, and the protein starts to unfold. And at this point here, you say you have the thermal melting temperature, the inflection of the curve. Another method that we use in the same instrument is DLS, dynamic light scattering. Here, light is scattered by the particles that are tumbling around in the capillary. In this case, it can be in a test tube. Here we want one peak. A homogeneous sample will give one peak, meaning that you have only one form of the protein tumbling around in the solution. When you see the red, like the red curve here, this means that you have something that is a lot larger. On this scale, we have the size of the particles. These things here are aggregated proteins. We don't want that in our protein sample. We want one peak, like this. Other methods we use at the DDD are size exclusion chromatography, STS page, and mass spec. Now, to the topic of today, SPR, Surface Plasmon Resonance Biosensor Technology. This is a technique that uses a tiny, tiny gold chip, seen here, you can see it for scale, the pin. On this chip, we have a glass support surface underneath, and on top of the golden chip, you immobilize your protein molecules, your targets. Then there is a flow cell above the chip. Here you flow in the compound, the ligand, also known as the analyte, the thing that is going to bind to your target protein that is stuck here to the chip. Then you shine in light, and the angle at which it reflects will change if you have something bound to the protein target. So you have one angle when you're flowing over a solution without anything in it. Then you have a refractive index and a certain angle here that is constant. Then you start flowing in your compound or it's called an analyte, and when it binds, the mass on the gold chip will change, and then this angle here will change, and that's what you record. And these are typical sensorgrams. They're called sensorgrams, what comes out of the instrument. This is with multi-cycle injection of an analyte, and you have a concentration series. This is the standard method. And then here you have what's called single cycle injection, also in a concentration series. On the x-axis here, you have what's called Roos, response units. And on the y-axis, you have time. Important concept of binding is how well do things bind to each other, and what you want to measure is the dissociation constant. That's what will drive which molecules you want to work with. Often the better, the higher the affinity, the better the KD. Those are the molecules you want to work with. Here we have protein and we have ligand, little Pac-Man, a little pie shape, and then this is the equation. So free protein, free ligand, at some point they will come together. You have a K on, an on rate and an off rate. And the KD is decided by the concentration of the free divided by the concentration of the complex. K on divided by K off. Now, I'd like to explain the SPR sensorgrams a little bit more for you. So here I am by my learning glass, which is like a whiteboard. If I start with the multi-cycle kinetics method, first of all, we have our surface, with the chip surface, where we have immobilized the protein molecule. Then we flow over a buffer, constant flow of buffer, and in this constant flow of buffer, we inject the compound, so we get a concentration increase. And that's what I will show here. So first of all, we have flowing along here, we have just buffer. Then at this point here, we inject the analyte. Then what happens on the y-axis is the response will increase. So something happens when the binding occurs, the interaction occurs. This goes up. And then here we stop the concentration. And the signal will go down again and back to baseline. So that was with one concentration, and then we do a concentration series. So then we have the second injection coming in here. The signal increases, and we stop it, and it goes back to baseline. The third concentration, increase in signal, down, back to baseline. Fourth concentration, and then fifth concentration. Like that, and then down to baseline. Now, if the molecules bind with one-to-one binding, then we can fit the equation to the data, to the curves, and we can get the KD from that. But we also always want to check the steady state plot. And that we do by taking points at where the compound has reached steady state with its targets, where the interaction is full for that concentration. And that's these points here. I'll take a new color. These points here. And these we plot in a sort of dose-response plot. We do that. So we plot the lowest constant. Now I have concentration here. And then I have my response here on the y-axis. So the lowest concentration gets a little cross there. The second concentration, the third concentration, and then fourth and fifth. And then we can fit a curve here. Like that. And what we want to do is that, or what we want the data to look like is that these points here should reach saturation. Then we know that we have saturated the binding at this concentration. And then we can, once we have saturation, we can determine the KD from here. And that's half that concentration there. So that will be a steady state KD. So up here, we have the KD from the steady state plot. Now, if you remember, KD is K off divided by K on. And K off here is the off rate. So that's how fast does the small molecule come off the protein. And K on is the rate how fast does the interaction happen here. In this plot, we're only taking these data points to determine the KD. But if we can use the whole sensorgram here, all the data, we will get the KD kinetic. Then we fit all the data to the equation. If the off rate is very slow here, if we draw a little mini curve here, it will go on, and then the off rate goes very slowly off. This means that the small molecule is bound to the target for a longer time. It's stuck to the target for a long time, and it doesn't want to come off. And this is something that we might want to include. If we find the compounds that do this, this could be something that's good for the project as a whole. This method here is the single cycle kinetics. Like so, we have again time, and we have the response on this axis. Here, this compound, or if it's an antibody, for example, has a slower off rate. And then what we do here is we inject one concentration there, one concentration there, one concentration. So here we're increasing the concentrations without letting them fall off and go back down to baseline. Then we get the increase in response like that. And then finally, we see that this off rate is slower. It could be even slower. It might be that it's as slow as that. And then we use all the data in the sensorgram to determine the KD for this interaction. And then we call the method single cycle kinetics. This plot, if we fit all the data to our equation, and what we get out then is the off rate and the on rate for the interaction. And from the off rate and the on rate, we can calculate the KD. If everything is great, then this KD will be the same as the steady state KD if we have reached saturation properly. Right, so as we saw on the learning glass, here are the sensorgrams. I wanted to show two different profiles that we had in a project where a set of compounds gave one type of SBR sensorgram profile and another set gave a different profile. So here we have one profile. Here we could take the steady state plot. We see we have reached saturation here. But also the fit of the equation to the data curves gives a sufficient fit of 1-to-1 binding, so we can measure or get out a KD for this compound. These compounds look like this, if I give a stylized image of it, with groups coming out in this direction. We instead look at this profile of the sensorgrams. You can see the off rate is a lot slower. These compounds had groups coming out in this direction instead. And again, the fit, with the 1-to-1 fit, is sufficient for calculating a kinetic KD here. So that's what the sensorgrams can be used for. Here you get two different types of kinetics, a slower off rate in this case, which could mean that you have a better compound. At least it's something that sits on the protein for a longer time. This might not mean that it's better in the long run, but it's a way of differentiating between the compounds. And now, so what can surface plasmon be used for in the drug discovery process? Why do we want to use it? We want the binding kinetics, the KD, the K on, and the K off rate. It can be used purely for affinity ranking. We have this set of compounds, we have this set of antibodies, which ones are best, which have the highest affinity. And that is used in the SAR. If we develop a compound in this direction, does the affinity get better or does it get worse? SBR can also be used for target engagement, purely showing that, okay, we have this compound from a screen, does it bind to our target? That we can say. And also, it can be used to validate the target functionality. Okay, we have this protein that is purified, it looks nice and homogeneous, but can it bind compounds that we know it should bind? These are the questions that we can answer with SBR. How does a compound bind to its target protein? My name is Annette Roos. I work with biophysical methods for drug discovery. I'm a crystallographer and structural biologist by training. So how can structure determination help the drug discovery process? I use a method called X-ray crystallography. Here we want the target protein of interest to be crystallized, to be able to visualize the atomic structure. We would like to crystallize the protein target together with the potential binders, the compounds that bind to it. We use 96 well plates with 3 little wells where we put our pure protein solution at pretty high concentrations. We have a crystallization robot to do that. It has tiny little pipettes here that will dispense small volumes. And then we aim for this. The protein should crystallize. We fish out the crystals with a tiny little nylon loop, 100 micrometers. And then we plunge it into liquid nitrogen to preserve it so it doesn't dry out in the air. Then they're sent to the synchrotron. The crystals are put into the X-ray beam. They diffract, and we measure the diffraction pattern. The diffraction pattern is integrated and scaled together. We have to solve something called the phase problem, and then we can get the electron density. And this is the data that we have to be able to determine the protein structure. There's a lot of maths in this step too. And then this is the problem. We have to fit the amino acid sequence of the protein that we have purified and crystallized into the electron density. And that then tells us what the protein looks like on atomic level. And here you can see I have placed phenylalanine into this electron density, and here's a tryptophan. And then this is what you tend to see in pictures of proteins, often ribbon or cartoon representation from blue to red. So blue is the N-terminus, red is the C-terminus, which is there. And then here we have a compound bound in the binding site. And what do we want protein structure information for? Well, we can design better drug molecules, better compounds, if we know how they bind. We can also get information about target engagement. Are the compounds we have that have given a hit in some kind of other assay setting, how do they bind to the target? Where do they bind? If we have antibodies as our drug molecule, then we can also do epitope mapping with X-ray crystallography. So there we get the structure of the antibody together with the target protein. That's visualized here. So here we have a Fab binding to the antigen, and then the epitope is the orange part, the interface between the two molecules. We can also do virtual screening. Then we look at the binding site. We take away anything that is bound there, leave the amino acids, and see what other types of molecules could fit in the same pocket. In structure-based drug design, we zoom in on the binding site and look at the compound that's binding and which amino acids are binding to it. And then from there, we try and change the molecule to get better interactions or more interactions with the amino acids in the binding site. And here, this picture shows the amino acids are in yellow, and then there are 3 compounds that are overlaid here. So it's 3 different structures that have been solved. You superimpose them, and then you can look at, are there any differences in how the amino acids are? In this case, there wasn't. And where do these molecules bind? Where are the differences in the molecules? And here's another way of showing this. We have the protein here, and then we zoom in on the active site here. And that's what the compound looks like when it's bound. This is a kinase, and this is sunitinib, a very well-known kinase binder that's bound here. Then we can see which interactions. Here we have interactions that are called the hinge binding region. Here I would like to first show what the electron density looks like. This is an electron density map of about 2.3 angstrom resolution. And here you can see the blue mesh is the electron density, and the yellow sticks here are the amino acids that have been placed into the electron density. So here you can really appreciate how well we can place the amino acids into the density. Here is an alpha helix. Comes along there. This is a so-called graphics program that is visualization of the electron density, and it's 3-dimensional. So you get a feeling of the depth. You can see things that are in the front and behind. Here is another graphics program that is more focused on the visualization of the protein. Looking at the electron density and the amino acids in the stick type representation or line representation can be quite, doesn't give you the full overview. So here's the same kinase colored the same way in this rainbow representation, again cartoon, from blue, the N-terminus, down to red, which is the C-terminus. And here is the binding site for this compound that is called sunitinib. We can zoom in there, and then here you can see more of the depth when I scroll away information. You can see more details. And here now some of the amino acids I have made into the stick representation, so you can see them more clearly. The other amino acids are now hidden. I can switch them on here. Now they're shown as lines instead. And I have highlighted amino acids that are actually in the binding site as this fatter stick representation. Then we can look at the interactions to the compound that we have crystallized this protein with. And we can try and see where can we grow the molecule to achieve maybe more hydrogen bonds. We can also visualize the hydrogen bonds like that. So here you have this hinge binding moiety. So you have hydrogen bonds here to what is called the hinge in a kinase. I can also read in a similar kinase, which I've done here, and overlay it. And then I can compare the binding site. Again, this is sunitinib bound in this binding site. I can compare what amino acids are interacting with sunitinib in this kinase. In general, they will be very conserved. Many residues will be the same, but you can find some differences. And then you can try, if you want to be selective towards a certain kinase, you can try and use the places where the two proteins are different. Where are the amino acids different? Try and grow out your compound so that it can interact with the different amino acids. Then you can achieve selectivity. So at the DDD, we tend to, if possible, crystallize our target together with the compounds that we produce, the small molecules that we develop. But we can also use all the information about structures that is available in the Protein Data Bank, the PDB. So this is a data bank containing thousands of solved structures where the scientists have published a structure and then deposited the coordinates and the data into this database. So I can download a structure from there and the data, check to see that I agree with what has been described in the articles, and then I can use these structures in my drug design as well to get input for how a molecule should bind, or just information about the structures that we're working on, the targets that we're working on, rather. So in the PDB, the structures there are based on experimentally recorded data. From the PDB, we've all heard of AlphaFold. So AlphaFold is a prediction program for determining protein structures, predicting protein structures from the amino acid sequence. So we can also use the models that are available via AlphaFold to get an idea of what our targets look like if there is no structure in the PDB database. Now, to summarize biophysical methods in a drug discovery setting, what we want to do when we're developing a medicinal compound, a drug, is that we want to block the natural ligand from getting access to its target, to the target that we're working on, the protein, for example. This is a numbers game. So if we consider the equation here, we have the protein, the target, freely in solution. We have the ligand freely. When do they come together? So the higher the affinity, the better they combine to each other, the more favorable that interaction will be. But it's always an equilibrium between being bound and not bound, being interacting and not interacting. So if you look at the pie shapes here, we have the red one is our drug-like molecule, and the green one here is a natural ligand. If there are a lot more of the green ones, they will compete with the red one. But if the red one has a higher affinity, it has a higher chance of sticking to the blue target. This is complicated in a cell situation, of course, because then you've got thousands of other things that are swimming around in the soup of the cell. A lot more disturbances. But biophysical methods, when we isolate the system in a test tube, then we can look at the binding between these molecules in an isolated setting. And if they can't be said to bind, then how can we say that they're doing that in a cell situation? That's a summary, but I would also like to talk a bit about the other methods that we do have for studying interactions. So we have a method called switch sense technology developed by dynamic biosensors. Here we have DNA levers that are coupled to a chip, and then we attach our ligand here. This is the protein target. Attach that with a little DNA strand, single-stranded DNA, attach it to this other single-stranded DNA that is stuck to the chip, and then we flow over the analyte or the other binding partner. It can be a compound, it can be another protein, it could be an antibody. This DNA strand that is stuck to the chip has a fluorophore on it. And when you get this binding event happening, the interaction between the target and the analyte, then the fluorescent signal can change. That's one way. This is called proximity sensing. Another way we can use this chip setting is to alternate the voltage on the bottom here. And since the DNA is negatively charged, if you get a positive charge on the surface, it will be pulled down. And the way this movement happens can be recorded via the fluorophore. Here, so you track how the fluorescent part is moving. And then if you have something bound here on top of the red ligand, the movement will change. It will alter depending on if there's something big here or something small. You can also detect conformational changes. So if you get a conformational change upon interacting with the analyte, then this signal can also change. This instrument can also be used in what's called a Y complex, where you have two different DNA strands with different fluorophores, a red and a green. You attach one protein on the green and another protein on the red side, two different coupling experiments. And then you add your compound here, which is a bifunctional compound. So it can bind to, one side binds to one side binds to the other. For example, when developing PROTACs. So PROTACs binds to the target, and then it will bind to the E3 ligase and brings the two together. And when you get the two interacting and pulled together, you get changes in the signal here. You get the kinetic rates for the ternary complex formation is what you say that you're measuring. Another method that we have is microscale thermophoresis. Here you have your samples in very small capillaries. You heat up with a laser. You heat one point in the capillary. The molecules will want to diffuse away from the local heat change, the raise in temperature in the point, and then you record how the molecules move away. So when there's no heat, they move and tumble in one way, and then when you add the heat, they will go away quicker. For this, you have to have your target fluorescently labeled or measure the intrinsic fluorescence by tryptophan. And then you add different concentrations of your compound or of your antibody, and then you get a dose-response curve from this, and you can measure a KD. Joining me now is Professor Helena Danielsson. Hi, Helena.
Thank you.
You were an early adapter of this new technology, surface plasmon resonance biosensors, using these in drug discovery. Mm-hmm. Yes. Tell me a bit about what it was like in the beginning.
Well, the first time I came in contact with this technology, it wasn't possible to do what I wanted to do because it didn't have the sensitivity, but I was still quite curious because I needed a method that could complement our enzymatic assays, because they weren't sensitive enough and didn't give us the information that we needed to drive our drug discovery projects. And the first time when we actually used the technology, we didn't know what we would be able to do. And we were sort of testing, can we do this? Yes, you can. Can we do this? Yes, we can. And then finally, we managed to set up the assays in a way where we really see that this is very useful information.
Okay, right. So what was the useful information?
Well, if you use a standard enzymatic assay, you will only get the Ki value, which corresponds to the Kd value. So it's only an affinity parameter. But the great thing with this technology is that you can actually get the K on and the K off. So if we look at this graph, where we have the K on here, the rate constant, and the K off rate constant here, and then we look at the diagonals here. The diagonals actually correspond to the Kd, because that's the ratio of K on divided by K off. Now, since it's a ratio, you can have on this diagonal, you can have a value with a certain Kd. This value has the same Kd because it's on the same diagonal, but here you see it has a very slow association rate, slow dissociation rate, whereas this one has a fast association rate and a fast dissociation rate. Now, if you want to develop a drug, where do you want to be in this graph? Ideally, you would like to be somewhere up here, right? Something binds very quickly, dissociates very slowly, has very high affinity. But in the beginning of a project, you may start out here or you may start here. You don't know where you are in terms of the affinity. And if you start here and here, you will think that this compound has the same characteristics, same kinetic characteristics. But the optimization process for this one should go this way. The optimization process for this one should go this way. So understanding how you can resolve the kinetics through this type of a method has been quite revealing. So I think this is one of the most important advantages of this technology.
Right. So how about now? What's happening in the field? What are the new things that are happening in the field?
Well, of course, the SPR biosensor technology that was originally developed became a really important breakthrough technology. And other types of technologies have come later to try to do the same thing. They become competitors. But and some of these other technologies, and also this technology that was originally developed, they become more sensitive, you have a high throughput and so on. But I would say that there are new additional technologies that work in a slightly different way that provide us with new information, such as conformational changes, and you can resolve more complex interactions. So if you really want to understand how a ligand can change the conformation of a protein when it binds, some of these biosensors are hugely important for this. And that's where I think that the technologies are becoming complementary, and you can really get more information about the mechanistic features of your ligands and how you should optimize them in order for them to be efficient and safe therapeutics.
Right. Conformational changes and mechanistic studies.
Exactly.
Yes. How about challenges that we face with our techniques?
The challenges are often related to the protein itself. Most of our work involves studying proteins. And we realize that the proteins may not be fully folded, or they might be unstable. So having the right form of protein that is functional over time is one of the challenges that we face. Another thing that is often a challenge is that when we start a project, we don't have any ligands that we can use for establishing our assays. So we can immobilize the protein and we can think that we have a nice sensor surface. But if we don't know that we have a ligand that reliably interacts with that protein, we can't really validate or optimize the assay. So getting the first ligand is very, very important. And a ligand for which we have other data that we can correlate the data that we're seeing from the biosensor experiment is quite important. So our struggle to find the first ligand is often the most frustrating thing in the beginning. Once we have that, then we have an assay, we can further optimize the assay, and we can compare new compounds with that original ligand that we found.
Yes, I totally agree. And some projects where we start, we don't have this reference, this known binder, so then it's really difficult to set up the assay and know if, do we have the functional protein on the chip? Yes. Yes, I guess to sum up then, what do we see for the future? We're still trying to understand how to use these methods. We know a good deal about how we're using them, but what would you say?
Well, I think in the beginning when we started using this, it was difficult for the medicinal chemists to understand why do we bother determining the kinetic rate constants, because they were really only interested in the affinities, right? Now I think with this type of a graph, it's quite obvious that we need to resolve the mechanism or the KD into the rate constants, but adding additional steps now for conformational changes. How do we convince the medicinal chemists that understanding the conformational change is important? Because we can't just simply give them a KD value and think that, oh, this is the affinity. The affinity will be for the actual binding, but the conformational change itself might be the most important part of the mechanism of that compound. So for us to communicate the complex mechanistic features that we can characterize with this technology, I think is one of the challenges that we have. But once we master that, I think this is a very, very important and very informative methodology that really can help us be more clever in how to discover drugs.
Right, thank you. So back to this slide. To summarize biophysical methods, how we use them in drug discovery. We want to block the interaction between the target and its natural ligand. We have many biophysical methods that can be used to detect this interaction. We mainly use SBR and then X-ray crystallography. SBR to get the affinity and to get the kinetics of the interaction, and then X-ray crystallography to look more into detail at the interaction. In the cell setting, it's a lot more complicated, but if we can isolate the interaction in a test tube, then we can get more information about it and know that we are on target. And I think that was all I would like to say for today. Thanks for listening.